Okay. All right, we're going to go ahead and get started. I want to thank everybody for showing up for Beer of the Scientist. It's been a long time. I see some familiar faces. So hoping to get more people out here today. I thought people would be excited to come out for another Beer of the Scientist. I get messages almost a couple times a week asking, when's the next Beer of the Scientist? When are you going to do Beer of the Scientist again? So, and then I go and I fly somebody all the way down from Canada to give a Beer of the Scientist, and we only got a few tables to show. That's all right. Anybody who's here, you guys are about in for a treat, and you guys are going to learn some cool stuff and hear from a cool guy. So hopefully, I'm going to try to do another Beer of the Scientist maybe next month. November, December are always kind of hard, so we might have to push till January. But so my name is Levi Beverly. I run Beer of the Scientist program. I have since 2014. We were out against the grain for a long time, and we've been here. Unfortunately, COVID kind of shut us down there for a bit. We tried to do some online stuff, but the whole point of Beer of the Scientist is to bring science to the general community of the city of Louisville. We present sometimes complicated topics in a way that we hope everybody can understand and enjoy. Today is not going to be any different than that. So the talk will be about 30 minutes long, and then we'll have plenty of time for lots of questions afterwards if you guys want to ask questions. So our speaker today is a really good friend of mine. We're actually postdoc fellows together at Memorial Sloan Kettering Cancer Center in New York City for some time. And then we were at the National Cancer Institute in Bethesda, Maryland for about a year together as well. So both of us trained as postdocs in the lab of Harold Varmus, who won the Nobel Prize in 1989 for discovering the genetic basis of cancer. So before Harold Varmus and his colleagues came along, there were two camps of scientists in the 1970s. One thought that all cancers were caused by viruses. One thought that cancers were caused by something else that was more intrinsic to your own body and your own DNA and your own genetic code. And Harold's work and his colleagues' work that they won the Nobel Prize for basically showed that it was changes within the DNA of your own cells that cause cancer. Okay, so that's kind of a little prelude into what we're going to hear a little bit more about today from Dr. Will Lockwood. So Dr. Lockwood now is associate professor at the University of British Columbia in Vancouver and a senior scientist at the British Columbia Cancer Center, BCCC. He's published almost 100 papers on the topics he's going to tell you guys about today. He runs many huge collaborative grants in Canada. He collaborates with lots of people in New York City still and across Canada and across the United States. So without any further ado, I'm going to go ahead and see if we can't get his talk going. And if we could just welcome Dr. Lockwood up to the podium, I'd appreciate it. I also say that Dr. Lockwood gave a seminar at the Brown Cancer Center earlier today to a room full of scientists as well. So he's had a long, busy day, and he's got to turn around and head back to Vancouver first thing in the morning. So good.
Yeah.
Okay.
Thanks so much. And I'm not going to lie, this one's a harder talk to give than the one I gave earlier today. So I hope I don't bore you, and I hope I don't overwhelm you with data. My intention's to keep it kind of not simple, but just give you an overview of kind of the approaches we're doing to kind of treat this really devastating disease. So Levi already introduced me, and I'm sure you guys know or are aware lung cancer is a really big worldwide problem. This is about Canada here because it's obviously Canadian, but the same holds true here and as well as worldwide. It's the first leading cause. The lung cancer is the most common disease now in men and women in many countries. Canada is one of them, obviously, and more Canadians die, and Americans too, from lung cancer than many of the other kind of cancer types combined. That's including breast, prostate cancer, bladder cancer, pancreatic cancer, all combined. So lung cancer is a huge issue worldwide that needs addressing. And I'm going to talk to you about that today. One of the reasons why lung cancer is so bad compared to other types of cancer is because many people are diagnosed when it's already spread beyond the lungs. So the term we call is metastatic, so it's already spread throughout the body and not just localized to the lung, which makes it extremely hard to treat. And I'll talk a bit about that later. So ways that we are going about kind of addressing this public health problem is in a few different ways. So I kind of listed some of the things that we find need to be addressed to get better outcomes from this disease. The ones I'll focus mainly on today, the ones that my lab specifically addresses, is trying to figure out ways that we can prevent lung cancer, and I'll tell you a bit about how we're looking at what causes lung cancer, specifically in never smokers, and how we might be able to prevent that. I'll talk a bit about lung cancer screening, and I'm sure people are aware of screening for other types of cancer, but lung cancer screening is something that's relatively new. I think BC is actually the only province that has it implemented on a public health, funded by the government on a population level. And then I'll talk a lot about the end, and which is what the main topic of my talk is about how we're going about developing better treatment strategies for people who have this advanced disease that most people come in with. So the main reason why lung cancer is so bad is because it's so common, but then almost everybody that gets it actually, unfortunately, dies from this disease. And you can see that this is the 5-year survival rate's around 15%, and these two bars are actually the change over about, I think it's a 20-year period, and you can see it's hardly shifted very much. So it still remains, despite new technologies, despite new treatments, we really haven't shifted that survival curve higher. And this isn't meaning that people are living a little longer, but we're not curing this disease, which is obviously our main goal. So why is the prognosis so poor for lung cancer compared to other types of cancer? And when we really boil it down, there's really two main issues for it. The first is that most patients, like I said before, have an advanced disease when they come in, so it's already spread within the lungs. And this is important. So down here you can see some plots. You can see that only around 15% are localized within the lung, and the rest of them are either spread regionally to lymph nodes or to other organs within the body. But this is really important because you can see that survival is much different depending on how this disease is diagnosed. So if it's localized, you have almost a 50% survival rate. And the reason for that is because if it's localized to the lung, you're actually a candidate for surgery. So you can actually get your tumor resected and often cut out your body so that it doesn't reoccur. So that's only an option for patients that come with localized disease. If it's spread within even the regional lymph nodes or the other organs, you can see that the survival rate drops dramatically. So one of the big things we're trying to do is, how can we make sure that patients come in and they're actually coming in when they have disease earlier so that they have this better survival outcome? So this is where lung cancer screening comes in, just like many other types of cancers. If we can catch it early, you'll have a better outcome. The problem is the lungs are within the body and they're really hard to visualize. You can't do a prostate exam where you can easily access, or we don't have a biomarker. It's not, for breast cancer, prostate cancer, this is really well developed. Colon cancer, really hard to do anything with the lungs. But recently this has actually changed, and this happened about 5 years ago, where these two large trials were done, one in the States and one by my colleagues in Canada, this Pan-Canadian Early Detection of Lung Cancer Study. And what they did is they actually had people who are at high risk of cancer come in to get a CT scan on an annual basis. So basically, they come in, we call it low dose because it's not as high, it doesn't give you as much radiation, so it doesn't have that effect if you come in routinely. And they can actually visualize the lungs and see if there's anything growing in there. And so this was actually shown to actually reduce the mortality rate by up to 20% in the population that was screened, which is a huge advancement for this disease. As I told you before, it's really hardly shifted over many years. So just to give you an idea what this kind of looks like, you can find these really small nodules. So you can find things that are only millimeters in size within the lung. So these patients will come in and they'll get their lung scanned, and what I've circled here is actually the node. So these are masses that shouldn't be in the lung. And there's some that come in, they're really, really small, and then they'll come in for another CT scan next year, and it still looks the same size. Next year, the same size. So these are really kind of indolent tumors, we call them. They don't grow very fast. This is somebody who you just watch. So they have a mass, it shouldn't be in their lung, but really going in and opening them up to do surgery really isn't needed there because it's a slow growing thing. If you look at this patient over here, they come in and they basically have a clean scan on the first one, and then they come in with a little mass the next visit, and then the third mass that's dramatically grown. So this is somebody who would be at very high risk that would get in right away for a resection of that tumor, or they cut out typically a portion of the lung or the whole lung lobe. So that would be a candidate for intervention. So this is really advanced kind of early detection and survival, like I said before. So currently, this screening is limited to what we call high-risk individuals, and this is really basically long-term, our current former heavy smokers. So basically people who are over the age of 55 that have smoked a lot either in the past and recently quit within the past 15 years, or they're current smokers, smoke a lot of cigarettes. So these are the patients. We can't screen the whole population because it's too expensive, so we have to find the high-risk individuals to get in for this intervention. So this is what it's currently limited to, but not all lung cancer occurs in smokers, so this is a huge problem. And it's a really big problem. I know Kentucky has, I think overall, probably a lot more smokers based on past history compared to where I live in Vancouver, where not very many people smoke at all. And in Vancouver, we actually see that a large fraction, and it's growing more and more, of the patients that come in with cancer are actually never smokers. So we've been really successful at smoking cessation programs. A lot of people quit smoking, which is great. So the proportion of cases, lung cancer cases associated with smoking is actually decreasing. In BC, these are probably a bit out of date now, but 45% of the people that we see in the clinics actually are former smokers. They've quit for over 15 years. And then up to 25%, probably now up to maybe about 35% of the people we see are never smokers, so people who've never had a history of smoking. And if you just took the never smokers that get lung cancer, that would actually be the 7th leading cause of cancer death in the world. So this is not just a disease of smokers anymore, and it's getting more and more prominent. So this is just kind of to show you here on the number of deaths responsible from cancer that never smokers is one of the very top. So now I just told you that screening can help cancer, but how can we identify patients that don't smoke that we should get involved in screening programs? And then we got to look at kind of what are the risk factors and what are the causes of lung cancer. Obviously, we know tobacco smoking is the #1 cause, but there are some other established risk factors from epidemiological studies and from other studies. We know that there's environmental exposure, so radiation exposure. Radon is a very big problem. I don't know, especially in British Columbia, which radon basically is a gas that from uranium decaying in the earth, and it often seeps into basements, and it's odorless and it's really hard to detect. So it's really hard to track radon exposure because there's no way to really tell if anybody's had exposure to radon. So it's a hard one to track, but we think around 10% of cases are involved with radon exposure. Arsenic isn't a big problem here, but in some countries it's in the drinking water. It can involve lead to cancer. And cooking fumes are a big one. So a lot of people who cook with oil and all that, especially in Asian countries, that is a huge factor as well that we know is associated with development of lung cancer. You don't smoke. Secondhand smoke was always thought to be a really big risk factor. We think it is, but it's being less and less kind of implemented now when we consider, less and less up the scale now when we consider all these kind of more recent things I'll discuss in a second. Viral exposure has always been thought to be involved as well, but again, it's been hard to track. And we know there's also a genetic component to this. So some people are more prone, families especially, to get lung cancer. So we know there's some genetic basis there as well. So we're actively looking for things that are involved in that. But what my lab's kind of addressing now are kind of more what I call emerging potential causes of lung cancer. So in British Columbia and many states down here as well, we've legalized cannabis smoking. Even though there's other methods to consume cannabis, 85% of people still smoke it. And now we have kind of this whole new exclusive class of people who we consider never smokers for the purpose of screening, because they don't smoke tobacco smoke, but exclusively smoke cannabis. So how could that be involved? We know that smoking took about 20 years to see the effects on people. So now these newly legalized substances now that people are reporting and maybe smoking at a higher rate, how can we figure out if that's going to cause cancer within 20 years? How can we be proactive about this situation so we can figure it out? The other big issue is air pollution. My colleagues just recently wrote a paper showing that air pollution is a tremendous risk factor for people who have never smoked. So we know that it's causally linked to lung cancer development. And again, that's not a big problem in some countries here, but even in Vancouver, where we have a pretty good air quality, we get this forest fire smoke quite often now. And it's a huge dose of air pollution at a very acute time. So we're interested in studying the effects of that kind of stuff as well. And so we can find people to get involved in screening programs. So who should we, how can we figure out more about this stuff to find individuals to screen? So kind of the way we're doing this is kind of a new experiment. We just got funded to do this. We kind of wrote a grant basically saying the Canadian government, you can't just legalize something without knowing the long-term effects. So in Canada, you can buy this. You can buy cannabis anywhere now. It's legal. There's no warning label on it or anything because we don't know. And all the studies that have been done to see if cannabis smoking is linked to lung cancer, they're mainly epidemiological studies, so they've tracked patients. But there's a lot of issues with what has been done in the past because it wasn't legal. A lot of people didn't report smoking it. The other issue was that almost everybody also smoked tobacco, so it's really hard to figure out what's the cause of marijuana versus the cause of tobacco. So we've kind of taken an approach where we can't wait for 20 years to get this epidemiological evidence. You have to be proactive and do it now. So we're doing this in a couple of ways. That's a big bag of marijuana there. It's a kilogram bag that I recently got. It's called Sour Cookies, which is apparently one of the more popular strains. So I wrote a bunch of cannabis companies and I said, what would be the typical kind of your best seller? Give me that. I want a kilogram of it. He said it. And then this big bag of weed showed up in my lab, and my graduate student looked like the happiest person in the world, and he had it. And then I told him, go roll some joints. We got some work to do. So basically, that wasn't a hard position to recruit for. And now what we got now is we got this smoking chamber where we actually take these joints that we roll, and it smokes just like a person would smoke. So you can actually fine tune the way it inhales the air just like a person would smoke. And it's a bit different. So we do this with tobacco cigarettes as well. Typically people smoke and they don't inhale that deeply with tobacco. In marijuana, it's a longer puff. They hold it for longer, so this type of smoke's a bit different. So we generate smoke using one of these chambers, and then we do two things. We can expose our kind of cells growing on a plate to them, or we can kind of make a condensate from the smoke and treat our cells. And what we're looking for there is a very basic question. Does it cause mutations like tobacco smoke does? Does it cause genetic changes to your DNA that could cause cancer eventually? And we think that if we can show that, we'll have a good idea of whether or not this could be a risk factor in lung cancer development. The other thing we're doing is actually looking for tumor development in mice. So we actually take these mice and take that smoke we puff from there and we put mice in a chamber and make them kind of smoke it every day for a little bit and see what happens there. So these mice eat a lot of food. My budget's gone way up, but it's kind of more long-term experiments. We're going to see what happens here. So this is again very infancy in the stages. These are the kinds of things we're trying to find, and it's all about what we should be asking people to get them involved in screening programs. So for the last little bit here, I'll talk about so that's screening and being proactive about health, which I think is the best way we're going to change lung cancer rates, survival rates in the near future. The other thing is we're still, even if we try to catch most of the cancers early, there's still going to be people who don't follow these criteria, don't get screened and come in with advanced disease. Okay, and what can we do about these patients? And this is where my lab has been focusing most of our efforts, and we're trying to find new ways for therapy. So this is going to be a little bit overwhelming one. I showed this one this morning too, but I just want to give you a kind of a scheme of what we're trying to do. So my lab uses a bunch of different what I call genomics approaches to kind of figure out new ways that we can treat lung tumors. So one of the ways we do it is what I call clinical genomics, and what I mean here is we're basically taking tumors from patients and then we do sequencing. So essentially what we're doing is we're looking at every single 20,000 genes in the human genome. We're assessing the genes to see if there's any mutations in them. And when we see frequently mutated genes, then we know that we have a potential cancer-causing gene that we can develop a drug for. Okay, so we're doing that, and that's kind of really good evidence that the gene is involved in cancer because we're actually looking at what happens in patients, right? One of the other ways we do this is we use mice again and cell lines. We call a thing called functional genomics. So what I mean by functional genomics is that we actually disrupt genes intentionally. So we can disrupt almost every 20,000 genes in the genome individually again and see what happens when we disrupt them. Do they cause cancer? And we can do this in mice and in humans. And I'm sure that some of you have heard about these methods called CRISPR and things that have really changed the way that we can do this. We can actually do this on a really high throughput scale to figure out genes that are causing, are involved in cancer development. And the last way is we actually just screen lots of drugs. We actually take panels of hundreds of thousands of chemicals and we put them on cells and we see which ones kill lung cancer and not normal cells. And then we try to work up what they're doing and how those could be involved. Thank you, sir.
You're going to be thirsty.
Yeah. So we use all this information to try to understand how lung cancer works and then you develop new ways for therapy. So I say this and I show you all the survival rates and it sounds like it's been really bad and we've made no progress at all, but this isn't true. This is my grandfather had lung cancer in 1971. He had cobalt therapy, which is really awful. Terrible therapy, doesn't do much. So we've made huge advances in terms of surgery, radiation therapy. Very standard things that have really improved patient survival. Chemotherapy, although it has a lot of off-target effects, has been proven to increase survival rates of patients, so there's been a tremendous advancements there. What we'll talk about a bit today is this kind of idea of targeted and personalized therapy and how we're kind of using that to treat patients. And then there's this whole idea of immunotherapy, stimulating the immune system to recognize human tumors. And lung cancer has been one of the ones that responds really well to this therapy. So some patients that are almost cured by getting immunotherapy, which we call. But I won't talk too much about that. If you have any questions about it, I'd be happy to answer it. We do stuff on that as well. But I'm going to talk about this idea of personalized therapy and how we're taking a new spin on it. So I'm sure most of you know, and this is just a really simple kind of schematic about how cancer kind of works in terms of lung cancer. So we get these mutations in these pathways. So this is what we call a pathway. You got a receptor molecule, which is on the surface of the cell. And then basically that, when it binds to this, what we call ligand or protein, that tells it to start signaling. It sends a signal that goes down through many other protein components, which sends information to the nucleus of the cell to tell it to grow and other things, right? So this is kind of how a normal cell would work, is it would turn on this pathway when it needs to do a specific function, and then it would grow for a little bit, and then it would shut off, right? That's how normal cell work. In cancer cells, we have an aberration of this process. So we get the same pathway, but now we get a lightning bolt here as a mutation. So you get a mutation in one of these proteins, and that tells it to not turn off. The signaling pathway just keeps on going, even though it should be turning off. And then you get too much cell growth, you get a tumor development, right? So this is pretty common. Schematic. So you get a mutation in one gene, but you can also get a mutation in a lower gene in the same pathway, and they can have the same net effect, right? So there's all these different genes that have similar functions when they're mutated. Essentially, the cell just keeps growing, doesn't turn off. So through all these clinical genomics approaches I told you before, where we can look at patients now and look at every single gene in their genome and look to see which ones are mutated, we actually have a pretty good understanding now of what drives lung cancer development. And these, don't worry about these names, these genes, but these are some of the mutations that occur in lung cancer that are known to cause the drive lung cancer development. You can see that we know about 55% of what we call driver gene mutations, and then we have about 45% of this pie, the gray area, which we still have no idea what's going on. These are patients that have been sequenced, but we don't know what genes are mutated. So those are ones we were really heavily focused on. But once we figured out some of these specific gene targets now, this led to this idea of personalized therapy now. So we know some of the genes that are mutated. Now if we can figure out how they work, maybe we can use that information to treat cancer. And this has led to this concept of personalized therapy. So this is basically the idea that you target an individual specific tumor depending on the mutation that they have. So in a normal cell here, I said that normal cell's fine, and this cancer cell, you have a mutation, right? Now the idea is if we can develop a drug specifically for that mutant gene, here's my drug, it binds to and inhibits that protein, right? Now it'll shut off that pathway and the cancer will stop growing. That's the whole idea of targeted therapy. So a patient will come in, they'll get their sequence of their genes, and if they have a mutation, one of these things, if we have a drug for it, we'll give them that drug, okay? And this has been a huge advancement for for survival of patients. Typically, I should have put the picture in, I had it before, but these patients come in, and our mice too that we developed, they can have tumors in their lungs, and they are all over the body, metastatic disease, and then they get one of these drugs that targets the mutations, and the cancer is like completely gone in a week. The whole cancer is gone. It's extremely amazing. Even our mice, they can't breathe because their tumors are covering their lungs, and then all of a sudden they're completely breathing fine the next day. It's absolutely astonishing. So they're really good drugs. The problem here is drug resistance. So this is a really nice schematic. It took me a long time to make this, so I hope you're that. So if we think about cancer pathways, my Ferrari here, okay? When the wheels are moving, the wheels need to keep moving for the cancer to grow, right? So the wheels are on, we're growing. So the drugs that we've been developing are kind of like putting on the brakes on this car. So we develop these drugs, and we put the brakes on, the car stops moving, pathway off, cancer dies. Okay? The problem is the brake pads wear out. So you treat patients with these drugs for too long, eventually, they just don't work anymore. The brake pads wear out, car starts moving again, right? And then you start, the cancer starts growing again, you get recurrence, and then a resistant tumor that ends up reoccurring. So how can we solve this problem of drug resistance and improve patient outcomes? It's a huge issue because these drugs work well, and how can we target resistance? Well, and I kind of segued to this, but if you look at these pie charts, I'm sure that you guys all know about pie charts. That this means that these mutations, when we looked at this pie chart, something never made sense to me, and it was, why don't cancers ever have more than one of these mutations in a gene. They're pie charts, so these are all fractions of patients. They don't overlap, right? They're all actually what we call mutually exclusive. They don't occur together. And this always fascinated me. I was wondering why do no cancers ever have more than one activating gene at a time? And if you think about it, you think that, this is our pathway. Again, if you had a mutation in this gene, and then you had a mutation in this gene, you think that the pathway would be more active and the cancer would grow even faster. So you'd think that cancer cells would actually want more mutations because they'd be more aggressive. And that's kind of what cancer is, just kind of like evolution, right? It keeps selecting for things that make it more grow faster and faster. So this never made sense to me, why this didn't occur. Why don't we see any patients that we sequence? We sequence hundreds, thousands of patients. We never see two genes mutated at the same time in these pathways. So we actually started modeling this, and what we can actually do, I won't bore you with the specifics, but we can actually engineer mice and cells where we can actually express both mutant proteins. So we can take ones that have an individual mutation and then make them express another one. And we just asked the question, what would happen if we did that? And when we did it, it was actually quite fascinating, is that these cancers actually blew right up. So if you look at this, it's just, don't worry too much, it's just, these are happy lung cancer cells here on the left. They look nice, they're all kind of nicely ordered, they grow really fast. These ones only have one mutation. But when you put the second mutation in, it's probably hard to see here, is that, but you can tell that they actually swell right up. They almost blow up. And if I zoom in a little bit here, maybe you can appreciate it a bit more, but they swell really, really large, and eventually they just kind of explode. And it happens really rapidly, it happens quickly, and it is a really fascinating way, because usually when we put the brakes on these things, they don't die like this. They actually kind of swell, they shrink up, and then they kind of die. This is like they're just exploding, which is kind of cool and not expected. So this is really interesting information for us, but you can see why this could be problematic. We can't make people who have tumors, we're not going to give them another oncogene, right? Like, we can't make them have another oncogene, or I don't think we'd want to. Obviously, you'd have a lot of issues with that, and not really effective strategy to employ. So we thought this is great information. We think it's interesting. That explains to us why we don't see cancers that have two mutations, but how can we use this information to better treat patients, which is what our goal is, right? So this kind of led us to this idea that maybe there's a new way to kill cancer. So we're always putting on the brakes. We established that. The brake pads were out. But now we have this kind of concept where we're actually flooring the gas pedal. So we're making these cancer pathways grow even faster. So we're just jamming it right on. So this car is moving like crazy now. And basically we're just overheating the engine, right? So now it's a totally different way. We're hyperactivating what we call the car or the pathway, and it's just exploding because of this, and the cancer dies. But how can we put on the gas pedal on cells? If we can't make them have another mutation, how can we put on the gas? And that's kind of what my lab's been working on. And then so that's what my lab's been working on, but first we kind of took it back again, and we asked, well, how do cancer cells actually avoid this in the first place? They're really smart, right? So if you have a mutation in this pathway, how do you make sure that it's not too high for the car to overheat? How do they keep it within that kind of Goldilocks zone where everything's just right and they can grow without overheating? So how this actually happens is very simple, and this is how normal cells typically shut off the pathway in a normal context. Is typically what happens is if you have a mutation, or if the pathway is turned on, what happens when it's turned on is that you get the response you want, so the increased growth. But then what turns on is this, what we call a regulator. So it's almost like a governor. It gets turned on only when these pathways get turned on. And then it goes back and it actually inhibits these pathways to turn it back off again. So what we question is whether or not, if we target this regulator now with a drug, and then we block the regulator as opposed to the mutated oncoprotein, whether or not that would take off the kind of this governing mechanism and then lead to this hyperactivation and overheating of the pathway. So what we've done is we've actually looked through using, again, backup patients to see which of these regulators seem to be commonly expressed in people that have mutations in these genes that we talked about before. And we've actually found a regulator that we're working on right now, and exactly that. We're lucky that we have a drug for it. We're screening for better and better drugs now. But I'd like to show you the cell picture again. So again, this is a different microscope, but same cells. These are lung cancer cells, happy ones with one mutation. And now instead of putting in that other mutation, now we have a drug for that regulator, one of those regulators I just talked to you about. And again, we see the same thing. And it's pretty hard to see here, but again, it's the exact same thing we're seeing. You can see it from some of these cells here in the middle, is that they swell right up and die. So it's the same thing we saw when we put on both mutations now. So we have a drug for a regulator then now we think hyperactivates the pathway and kills the cells. So just to summarize how this works, so we now, again, our car, right? So we have an oncogene drug. That's the typical way we've been treating cancers. You come in, you get sequenced, you get a drug that shuts on, puts off the brakes. And now we have this idea of this gas pedal. So if we put the gas pedal on, the car overheats and dies, and we can do this by using a regular drug for one of these regulator proteins. So it's basically taking off the governor of this system, so the whole thing overheats and dies. And the cool thing is that not only do we think that these regulator drugs will work in the context of people who come in with primary disease, so no untreated disease, we actually think it works really well. We've shown this. They actually work in the drug-resisting scenario as well. So we think the patients that get the oncogene drug first might come in, they have really good responses at the beginning, but once they develop resistance, then we could give them this regulator drug and hopefully prolong their lives for even longer. So we think it could work in both contexts and circumvent this major issue that we've been trying to address. So Levi will probably get mad at me if I go too much over time, so I'll just summarize. I hope I've shown you today that lung cancer is a major problem, and it's not just a smoking disease. It's a very major and prominent problem among never smokers as well. I've always shown you that screening can improve the outcomes of lung cancer patients, and if we can figure out ways that we can get more and more patients involved in screening that are at high risk, it'll be effective. Current strategies that are used to treat lung cancer can be effective, but they often suffer from this disease resistance and relapse. And for the first time, we've actually found that killing lung cancer cells through this activation or hyperactivation of pathways could be an effective means to treat patients. And our next step is to translate this into clinical work. So we're actually looking at other cancer types, and we think this could work in pancreatic, glioblastoma, colon cancer, and we actually are working with companies now to develop new drugs that we think could be useful in patients. So we're hopeful that we'll be able to advance this in the coming years. And I get to talk about stuff, but these are actual people that actually do the work in my lab, so I like to show them there. And importantly, which most people will tell you, is we can't do any work without funding. So I think Levi likes, or he's big on us telling people what we do because we need to tell people what we do. You know, we get funded by many different sources, and if we can't explain to you why what we do is necessary, we won't get funding, and we need it to do the work we do. So we always appreciate our funders as well. So thank you very much. Happy to take questions.
So we have plenty of time for questions. One thing I want to kind of point out too, that Will's saying that lung cancer is a disease of non-smokers as well, and a lot of times I find myself, I'll talk to people and they're, oh, my dad or my grandfather or whoever, my father actually passed from lung cancer a couple years ago. First question I usually ask is, were they a smoker or not? And it's not because I'm judging you that you deserve to have the cancer, but because some of the things he talked about is, I can almost infer what kind of therapy you might go on or they might go on based on their smoking history. Because the one mutation that he was talking about is much more common in non-smokers. And these targeted therapies are used much more in non-smokers. So it's not about like, oh, I'm not asking, did you smoke, did they smoke, did they not smoke because of that. I'm asking because now I could probably almost guess what therapy they're going on, because the targeted therapies have, not only are they very effective up front, they have almost no side effects.
Yeah.
Whereas if you're a smoker, you probably have one of the other mutations and you're going to go on chemo, radiation, that's nasty shit. So.
And immunotherapy actually works a lot better in smokers too, so that's a common thing now. So it does have some, yeah.
So we have any questions? Yeah, Robert?
Yep.
So Louisville doesn't have the best history for air quality,
Yeah.
although it's gotten better in the last 15 years or so.
Mm-hmm.
Is there any correlation between things like power plant and industrial pollutants and any specific mutation or any?
That's exactly what we're doing now. So my colleagues have recently, so the hard thing is finding people who have been exposed to, like high levels of pollution over time. So what they're doing and what they did in the past is they can actually use now satellite imagery and residential history to kind of infer how much pollution you've been exposed to. The question there is whether or not pollution in different areas of the world is the same. And I think that's a really important question, if it all acts the same way, and what is the components of the pollution? So that's a question that we're working on. We actually just we're working on a study right now where we can use this new method to kind of take never smokers now who have been at a high risk or high cumulative pollution exposure and no pollution exposure or low levels, and now directly compare the mutational signatures to find those genes that you're talking about. So that's the question we're actually looking at, because like Levi said, a lot of these kind of etiological agents are associated with different mutational types. We think it might be as well. So this is a study that will actually first tell us. But the problem has been defining people who have had high exposure versus low exposure before. And obviously you can't have smoking in there either because it could confound things. So that's what we're trying to do, this study like that. Yeah, it's a really good question.
Yep.
Yeah.
Hi, sorry I walked in a little bit late with my wife and my 2-year-old. I can't hear very well, so I'm walking up here. Levi, foremost, sorry to hear about your dad.
Shit happens.
Yeah. So because we walked in a little bit late, I guess I missed a little bit of the thesis of the part of the argument in terms of the connection, like the cause and effect between marijuana and lung cancer. So what's the verdict? I mean, so does pot cause lung cancer? I mean, obviously there's a strong connection between tobacco and lung cancer, right? We know that, right? The smoking and whatnot, but does smoking cannabis have the same level of risk? Or
it's essentially the question that we're trying to address right now.
Okay.
So I was explaining it. Well, we don't know.
Oh, okay.
Yeah. So
Probably won't know for 20 years.
yeah,
Okay.
but so we know to answer some of your questions. I think that we have looked at the components of smoke, cannabis smoke versus tobacco smoke, and most of the same carcinogens that we know cause cancer within tobacco smoke are in cannabis smoke as well. The main issue here is whether or not people don't smoke as much cannabis as they do tobacco. So whether or not the
It's also
levels,
unfiltered though, too.
but it's also unfiltered, and they also smoke it very differently. So we don't know how by holding it in and breathing deeper, but the same carcinogenic components are in it. It just has not been shown to cause, say, mutations, which we are working on right now, or linked to disease because the epidemiological information isn't there yet. So we're treating mice with cannabis smoke to see if that occurs. We're gonna have to wait a couple of years before we figure out the end result. So.
Well, and I always tell people that anytime you combust anything and inhale it, it's bad.
Yeah.
Period. It doesn't matter what it is that you're combusting. It's bad.
Thanks.
Yeah.
Now we've got chew products that are bad too, right?
Yeah.
Chew?
They
Yeah.
cause cancer.
Oh yeah.
Well,
Tobacco chew.
Chew, yeah. I mean, some people would argue. Well, again, it's like levels of bad, right? Like smoking,
It's gotta be
chew,
different
vape.
Different things.
It's different levels of different
components.
Yeah.
It is different. So the interesting thing. So chewing tobacco it's different, and that is one of the things I get asked a lot. So it's actually quite interesting. So nicotine products, like things that contain nicotine itself, gets broken down into carcinogens that cause cancer. There's actually one of them that we use commonly in the lab. It's called NKK, but it's a derivative. It's basically a metabolized nicotine product. And if we take just that, and we actually inject it in the stomach of mice, they just get lung cancer. So that's a component that So it actually causes cancer through nicotine products, which I think you're ingesting, which is the same kind of idea. That's not going to be present in cannabis, right? But there's many other things in tobacco smoke that are carcinogenic. So you're right, that's a different route of disease, but I think so we don't know, but there could be equivalent in cannabis as well. We just don't know yet. Yeah.
But what other routes are there? I mean, as you say, they're different routes.
Yeah.
So have you looked at all the various routes? How many different routes have you looked at?
Like for consumption?
Individual chemical packs. Okay, each of these are chemical.
Oh yeah, right. So the smoke itself has a ton of different, the composition of a bunch of different chemical components. We're just testing it as a whole right now. After we'll go through and test them individually is the idea. So we're just doing it would take us forever to test all of those separate ones. So we're just taking the whole component, like the smoke itself, and just treating cells and mice with it.
So has this been done on tobacco?
It has for some components, yeah. So we're directly comparing cannabis and tobacco in these experiments we're doing. Yeah.
So one of the experiments that they're doing is they're taking the distillate, so they're taking the smoke and basically like turning it into something that you can put on cells. But
Yeah.
these were done in the 1950s with tobacco smoke,
Right.
and it was put on like rabbits' ears and rat tails, and shown that you can take this distillate and you can put it on an animal and it will cause cancer.
Yeah.
So
I'm very familiar.
yeah, okay. So one of the things that I've actually gotten the comment before, like, nobody's ever actually shown that tobacco smoke causes cancer, and I'm like, bullshit. I mean, they showed that in 1903 by putting these things. All right, it's just So These are the kind of things that have been done with tobacco, have not been done with cannabis yet.
Yeah,
But-
so our goal with cannabis isn't like not to do it or anything. It's just what we need to know so we can be proactive about health. Like I was telling you, prevention is a huge thing. Early detection is a huge thing. So even if it's like a warning label is our end goal here, or just getting people that smoke a lot of cannabis to ask the question and maybe get enrolled in a screening program is kind of what we're doing.
Then you got the other thing is the radon, which has
Yeah.
no chemicals whatsoever. So what's it?
So it's radioactive gas, yeah. So it basically causes direct DNA damage when you inhale it, and so it causes
So there's got to
mutations.
be a chemical route there, a chemical entity.
So you inhale the gas, and it just directly destroys your DNA, and then you end up getting mutations over time. So it's kind of how it works. Yeah,
That's a direct effect.
direct effect. Yeah.
Yeah, question back here?
Yeah. So if you were to have those risk factors and you wanted to get involved in like a study, do they have them here in Kentucky?
They do, I believe. They're kind of more trial based, and I don't know if your health insurance covers it. I guess the beauty of being in Canada is that we get it. It's all universal, so it's easier to implement. I imagine it might depend on your health insurer. I actually don't know. My colleagues have actually made what they call like a calculator thing where you actually like fill out a survey and it tells you if you should be enrolled or not. I just tell everybody to ask the physician because I think the questions that you got to ask, and it's proven to improve outcomes so much that if you know of anybody that's high risk, I tell all my family members who used to smoke and all that, you should ask your doctor if you meet the criteria and get involved, right? So worth the question for sure, but I don't know how it works down here. We're a little bit different.
I mean, I'll say here in the state of Kentucky, essentially the county, so it's usually countywide stats that we have. And so the counties that are obviously east or coal mining and all that stuff, like they have much higher incidences, but you can almost overlap lung cancer incidence with household incomes and with education levels. And because of a number of reasons, like they don't have a lot of access to healthcare in the eastern counties. B, they just don't want to have access sometimes, and B, they can't afford to have access. And so, unfortunately, that you come up with these other socioeconomical issues that can't be easily overcome either when you talk about screening and prevention.
And those are actually part of the questionnaire to get enrolled in screening programs. A lot of socioeconomic stuff, diet, and a lot of things like that.
Yeah, right here.
Comment on the surge in vaping and the connection there.
Yeah, I get asked that all the time too with vaping. Again, it's one of these things that I was talking with Levi's graduate student about it today. Is one of these things that we don't know because it's probably going to take quite a few years before we find out the effects. Do I think vaping is going to be different than smoking cannabis? I think it probably will be. I think that's probably we're starting out with the smoke, and the next thing we can do with these exposure things and all that is the vaping. So I mean to answer your question, I don't know. I think it would be different, but
again, combusting anything and inhaling is bad, but there's a scale of bad.
yeah.
And I feel like maybe vaping might be lower on the scale of bad, but it's not good. So,
Then you have ingestion in food.
Yeah.
yeah, I mean, unfortunately, like we always say, like, if we can eliminate all smoking of anything, better diets and something else. Like we could eliminate probably most cancer, 50% of cancers, but we're not going to all do that. So alcohol. Oh yeah, I forgot about that one. We're not going to eliminate that one. So.
Yeah, we don't know about ingesting. You're right. It's a good point too. So
Yeah.
Apart from the CT scan, are there any other screenings that you can do to detect it?
For lung cancer?
Yes.
Unfortunately, people have looked at a lot of different things. So the answer to your question is no. It's the only one for screening now. People have looked at a lot of different things. So kind of taking samples of your milk cells and using that as a risk factor. One of my colleagues actually is working very heavily on breath. So breath she actually has a study where she showed that she could diagnose COVID with breath because we don't really know how it works yet. We don't know if it changes kind of what we call microbiome, the bacterial components of your lungs, and it gives off different volatiles. But it basically your breath profile changes depending on your lung function. And so right now she's currently looking at people with and without cancer to see if she can come up with a breath signature that would be indicative of early stage cancer. So I think that one's probably pretty attractive actually. The data looks quite good and hopefully that'll make access to this a lot easier for people. if we can get it going. But that, and we're working on genetic testing. So we have some genes that we're interested in that we think cause the familial aspect, and our idea would be like just like breast cancer, where there's genes associated with risk. That would be a genetic screen that you would then go in again for screening on a proactive basis, just like in breast cancer. So I think there's emerging ones, but nothing I don't see this.
And I'll just throw out a plug for U of L also has a group that's working on breath analysis as a potential. Yep,
Are
Robert.
y'all doing anything with PFAS or PFOAs? Have you looked at that at all? PFAS, the perfluoral alkaline substances, and
the PFAs as well?
yeah.
No, we haven't looked into that at all, but yeah, good questions too. Yeah.
What about liquid biopsy?
Liquid biopsies? Yeah, I mean, liquid biopsies, I think for these drug-resistant tumors, I think trying to figure out secondary targets, they're going to be effective as well, I think.
Can you detect it through liquid biopsy?
So that's a bit difficult question. I don't think that's too expensive to do in a kind of a detection setting. I think, but what people are using kind of that liquid biopsy strategy for is to actually measure recurrence of tumors.
Anybody doing that in the U of L with a liquid biopsy?
Yeah, like circulating tumor DNA.
Yeah.
Yeah. Mm-hmm. Yeah.
Actively or just curiously?
Yeah, not sure. I mean, I've heard about it. I just don't know if it's like they're trying to pursue it clinically or,
Yeah.
like you said, curiously.
Okay.
So Will,
Yeah.
I was thinking like the incidence of lung cancer in United States is much more comparatively to tobacco-related other cancers, like head and neck cancer and all. Because my whole PhD topic was on lung cancer and head and neck cancer.
Yeah.
So I screen lot of patients'
Mhm.
blood and trying to find out the biomarkers, correct, like SNPs and all kind of stuff. And I used to collect the blood from the same family having the blood relation, like the brother or something.
Oh, yeah, okay.
So it's funny, like they were asking, why you want my blood? I'm not the patient.
Yeah.
But my question is like, definitely tobacco causes cancer, but I think there are much lesser incidences in United States compared to like Asian countries and all, because tobacco chewing is more common in, like, let's say from India, correct?
Yeah.
So I think same product, same pro-carcinogen or carcinogen tobacco has. So I saw like many throat cancer patients or like tongue cancer
Hmm?
related to tobacco, but I don't find any kind of patients relatively less here. So what you think, like that is associated with smoking, or like it's like definitely chewing and that are two different things.
Like
Same components, but
the mechanism is different, you mean? Is that what you're saying?
means like I think high incidences are more in Asian countries.
Oh yeah, right. And I mean, I think that has a genetic component, right? Like yeah. So I think it's we don't diet. Even diets in different different ancestries have different effects too, right? Associated with onset of disease like esophageal cancer and things like that. So yeah, I think you're definitely right.
Yeah.
But is it still true that, especially in China, rates of non-smokers that get lung cancer are higher, but that's because of the cooking that you're talking
so
about?
The cooking's a big one. But
oil.
even in Taiwan, they just did a recent big study and of never smokers, even for kind of controlling for all the cooking and all that. It's still higher in never smokers. The pollution was the big one that came out of that. Yeah. Nitrosamine from food products was another one that was implicated in terms of never smoking lung cancer.
Yeah.
Yeah, a quick question. You mentioned earlier about how, like, in the early 20th century, the tobacco industry was telling you that smoking was good for you, basically, right?
Yeah.
This maybe like in the 70s. So I'm wondering, like,
9 out of 10 doctors prefer Camel.
Yeah, no, that's right.
Yeah.
That's right.
Yeah.
So, like, is the cannabis industry involved in this kind of stuff in the same or similar way? This may be a social scientific question, but I'm just not calling into question, the quality of your research in particular. But I'm just curious about their level of involvement.
They're not involved at all. I've actually
Philip Morris funded studies were basically
Yeah, so no, they do not fund any of our work. And
No, I mean, generally speaking, though, like.
No, you know what? Honestly, they should be. I've used the argument before that they should want to know this answer because they don't want to be at the same place that tobacco companies are later. But I think for them, ignorance is bliss right now, right? They don't want to know the answer either, but they should be more proactive about it too because you know, in 20 years maybe we'll have lawsuits against them too, right? Who knows, right? So,
Right.
yeah.
Thank you.
Yep.
So with the cooking oil, when it's smoking, that's when it actually smokes. That's when it's supposed to hurt, right?
Yeah.
So if you're using oils with like a higher smoke point, is that supposed to be better or?
I'm not going to try to. I don't actually know the answer to that question, but I would imagine it could influence it. That's a good question. I should find out more about it. And types of oils do vary depending on where where people cook. So it's a good question. I actually don't know the answer.
I would think the higher smoke point would make less combustion probably at the lower temperatures, and so it's probably better. But again,
The regulator drug is to kill the actual cancer cells. So if you don't stop the initial whatever stimulating that as well, would the regulator drug potentially be a maintenance drug where it's still producing the cancer cells? It's just you're exploding them basically.
So so that we've actually talked a lot about this today too. Is kind of when to use both of these. I think is a good question. I didn't talk about it today, but we actually have some weird phenomenons of the strategy now is kind of one size fits all. You come in, you have this mutation, you just get put on this drug, and you just keep taking it, right? But that might not be the most effective way. And how you actually kind of use drugs, how the timing is something that's really not talked about much. Like, should you always be on it continuously, or should you get it for? Maybe you should alternate between these drugs over time, and maybe that'll prolong the kind of this resistance thing that we're talking about. So I think these are questions because these drugs work really well. I think it's a matter of just trying to figure out how to use them in the best way to prolong life. And I think that if we get more clever, but it might be very individual based. It might be dependent on how advanced your cancer is when you come in. All these other issues and then maybe your dosing strategy will change accordingly. So I can kind of see us going that way with some of these drugs. And then the regulator drug would fit into that, I think, too, is whether or not you put them on both at a time and then take one off, or what order you do it, and that might exploit this hyperactivation a little bit better as well. So it's a good question.
Yeah.
Is there a crossover in the mutations that you mentioned that occur in other types of cancers? Those specific to one?
Yeah. It's so the EGFR mutation I talked about. Let me go back here. Answer your question is some of them are specific for lung cancer. You don't see them really in any other diseases. Other ones are common to many cancers. So KRAS is one that I talked about a bit, or I showed. So is KRAS one that's about 25% of all lung cancer patients? That happens in a lot of cancers, and usually all of them are high fatality cancers. So pancreatic cancer, colon cancer, melanoma. That's a common one across cancers. This EGFR mutation that's in about 15% of patients. Well, Levi was saying it's commonly associated with never smoking lung cancer. It's very common in Asian females that get lung cancer, and it's exclusive to the lung. You basically don't see that type of mutation anywhere else. So some of them are common, some of them are not.
So when you talk about no cancers have mutations in more than one activating gene at a time, is that something that we've just observed, or is that like a provable thing that it can't exist?
Well, so this has always been known, I guess, but the explanation for it was kind of just that, well, they all do the same thing, so you wouldn't need more than one. And then until we proved it recently that if you actually do express both, they don't die. So the idea there is that if a cancer cell had one of these mutations, it's growing in the body, and then all of a sudden it gets this other mutation, that cell would die. So then it wouldn't form any of the tumor later on. So the idea is that it's not that these don't occur, it's that we don't see them at the end when a tumor develops. So they probably occur all the time. They just, since it's lethal, they die.
So I would say that, just a funny story, Will was telling me this, and I remember this when we were postdocs together, and he and the other guy that did a lot of this work, basically this idea, they were sitting around the break room drinking coffee, talking about so why don't EGFR and KRAS mutations ever occur in the same tumor? And everybody's like, oh, it's because they're in the same pathway, so they do the same thing. And they're like, that doesn't make sense, And I just remember these guys just drinking coffee talking about this, and then they go and do the actual experiments that like changes the dogma in the field, right? So it's like sometimes drinking coffee or beer and having a conversation about science will change the way that everybody thinks about a particular thing in cancer research, and sometimes that's how it happens. So I saw, yep.
So what next step are you most excited about?
There you go.
This is regulated drugs is my biggest thing. So we've actually, like I said, we've started kind of a company or working, and we're trying to develop these drugs now. So I think that's my I think what I have the most hope for going forward is I really think, like Levi said, this is something that this whole idea of why we did this in the first place is something we could never get money for. So if I tried to explain to you why I wanted to do this, I wouldn't have a good answer for you. It would be hard to get funded. Pretty much everybody say there's a waste of time, like where are you going to take this? But sometimes in science, these kind of questions that we don't really know where they're going to lead us. And for me, this was a really kind of basic question that I just wanted to answer. And then it led to what I consider a whole new class of proteins we haven't considered targeting in cancer yet. So this could lead to a whole new area of drug discovery that we haven't explored. It could lead to a new whole repertoire of drugs that could be useful in different types of cancer. So I think that's what I'm most excited about. But I like to use that example because it would never get funded. And sometimes these things that, I always see like people, certain senators around here, I guess, that maybe actually in your state a lot of times, that send things like NIH grants, some grants that we're working on. The description of like, why is the government giving money to this guy to do, study why elephants don't get cancer or something like that, right? But you don't know where it's going to lead you. Essentially, sometimes these kind of basic questions end up leading to something that's actually very relevant later on. So yeah, I like that example. That's why we got to fund basic research. Not everything has to be clinical.
So what effect does the regulated drug have on non-cancer?
That's a good question. Very good question. So yeah, you're right to ask that question because the idea there would be if that regulator was disrupted, you'd probably turn on the same pathway that causes the cancer, right? So maybe you'd be giving cancer to people if you treated them for a long time. So a couple things is that these regulators aren't actually very express. They're not really present in cells unless they have kind of really high activation of these pathways. So for one, if you use this drug, normal cells usually won't even have the target that it disrupts. So again, that's kind of going to selectively kill the cancer cells. The other thing we think is interesting is that it's really acute. So when we treat cells with these drugs, they die right away. So we don't envision a scenario where we'd be treating patients for a really long time so that they'd have the off-target effects. We think they'd probably be pretty effective right off the bat. So it's a really good question. I get asked that a lot. And make sure you don't give cancer to patients when you're trying to cure it, for sure.
So now I'll just end with one other quick little story, that kind of what Will was just saying a couple seconds ago. I was sitting in a conference and a recent Nobel laureate was giving this talk, and he was talking about the work that Jim Allison, the work that basically he won the Nobel Prize for, and he said that the work had nothing to do with patients. They were just curious about why cells acted a certain way. And then that he ended up winning a Nobel Prize for this work. And that's the basis of these immunotherapies that Will mentioned very briefly, and that are now curing patients that were incurable 5 years ago. So you just never know when, like, sitting around a conference room talking over coffee about some basic biology question might be the next big discovery. And with that, I would appreciate everybody, give Will a round of applause. Thank you all for coming, I appreciate it. And now, Will, you're off the clock, so thank you very much. I appreciate it. That's